There is something that can destroy your brain. The infectious agent has no DNA, no RNA. It's not a virus, not a bacterium, not a parasite. It's a single protein, folded into the wrong shape. You can't kill it with standard disinfectants. You can't kill it with radiation. You can't kill it with heat, because it was never alive to begin with. And if it gets into your brain, nothing in modern medicine can save you. The fatality rate is 100%. This is the prion, the most terrifying molecule in biology. Your body runs on proteins. They're the molecular machines that do everything: carry oxygen, build muscle, digest food, fight infection. A protein is a long chain of amino acids that folds into a specific three-dimensional shape. The shape is the function. A hemoglobin molecule folds into a pocket that carries oxygen. An antibody folds into a Y shape that locks onto invaders. If a protein folds wrong, it usually just gets recycled by the cell. Misfolded proteins happen all the time. Your body deals with them, except for one. One misfolded protein doesn't get recycled. It does something no other molecule can do. It teaches. Every human brain contains a normal protein called PRPC, prion protein, cellular form. It sits on the surface of your neurons. We're still not entirely sure what it does. It may help with cell signaling or copper transport, but we know exactly what happens when it folds wrong. The misfolded version, PRPC for scrapey, doesn't just sit there. It touches a healthy PRPC molecule next to it and forces it to refold into the wrong shape. That newly misfolded protein does the same to the next one, and the next. It's a molecular chain reaction. One bad fold converts thousands, then millions. Like a row of dominoes that never stops. No genetic code, no reproduction, just geometry, spreading. As misfolded prions accumulate, they clump into insoluble plaques, dense tangles of protein that neurons can't break down. The plaques kill brain cells. As cells die, they leave tiny holes. Over months, the brain tissue fills with microscopic vacuoles, empty spaces where neurons used to be. Under a microscope, an infected brain looks like a sponge. This is why prion diseases are called transmissible spongiform encephalopathies. The damage is irreversible. There is no treatment. There is no drug that can refold a prion back to normal. The disease progresses from memory loss to personality changes to inability to move to death. The average survival after symptoms appear is about 1 year. Prion diseases appear across species. In cows, it's BSE, bovine spongiform encephalopathy, or mad cow disease. In sheep, it's scrapey, named because infected sheep scrape their wool off against fences. In deer, it's chronic wasting disease, currently spreading across North America with no way to stop it. In humans, the most common form is Creutzfeldt-Jakob disease, CJD, which strikes about 1 in a million people per year, usually spontaneously. But the most haunting human prion disease is Kuru. It was discovered in the 1950s among the Afar people of Papua New Guinea. They called it the laughing sickness. Victims would laugh uncontrollably as their brains deteriorated. It spread through funerary cannibalism, eating the brains of deceased relatives as a ritual of respect. Here's what makes prions uniquely terrifying. Every method we use to sterilize, heat, UV light, radiation, chemical disinfectants, works by destroying DNA or disrupting cellular machinery. Prions have no DNA. They have no cellular machinery. They're just a shape. Cooking meat to 600 degrees Fahrenheit doesn't destroy them. Autoclaving, the gold standard for sterilizing surgical instruments, doesn't fully eliminate them. Formaldehyde, which preserves dead bodies, doesn't touch them. Even burial doesn't work. Prions can persist in soil for years and remain infectious. The only reliable decontamination requires soaking in concentrated sodium hydroxide or bleach for over an hour, or incineration at extremely high temperatures. We're not fighting a living thing, we're fighting geometry. But there's something else spreading right now that might be even more dangerous. Because prions resist standard sterilization, they have been transmitted through contaminated surgical instruments. In documented cases, patients developed CJD years after neurosurgery performed with instruments that had been used on an undiagnosed prion patient, even after standard cleaning. Today, instruments used in brain surgery on suspected prion cases are destroyed, not reused. Prions also contaminated human growth hormone supplies in the 1980s. Before synthetic growth hormone existed, it was extracted from the pituitary glands of cadavers. Over 200 people worldwide developed CJD from contaminated hormone injections. The incubation period? Up to 30 years. Chronic wasting disease is currently the most pressing prion crisis. It's been detected in deer, elk, and moose across more than 30 U.S. states and 4 Canadian provinces. Infected animals shed prions through saliva, urine, and feces. Those prions bind to soil and plants, remaining infectious for years. Healthy deer eating contaminated vegetation become infected. There is no vaccine. There is no treatment. There is no natural predator pressure sufficient to eliminate it. And the question that keeps researchers awake, could it jump to humans? So far, there's no confirmed case. But BSE, mad cow, wasn't supposed to jump to humans either, until it did. The challenge is fundamental. How do you design a drug against a shape? Researchers are exploring several approaches. Anti-prion antibodies that might block the misfolded protein from contacting healthy ones. Small molecules that stabilize the normal PRPC fold, making it resistant to conversion. Gene therapy to reduce PRPC production entirely. If there's less normal protein available, the chain reaction slows. In 2022, a woman with a genetic prion disease began receiving experimental antisense oligonucleotide therapy, molecules designed to silence the gene that produces prion protein. It's the first treatment of its kind designed to silence the prion gene in a living patient. Results are still emerging. A single protein, folded wrong, can destroy a brain. It can't be burned, it can't be bleached, it can't be killed, because it was never alive. And somewhere right now, in a deer in Wyoming, in a lab in London, in a brain that hasn't shown symptoms yet, it's folding.